Okay, thank you very much for coming, everyone. It's my pleasure today to welcome Cameron Ghalambor to give this week's Museo de Ciencias Naturales research seminar. So Cameron is an evolutionary ecologist working on adaptive evolution and the patterns of biodiversity that it generates. He's published papers on a whole range of areas that are interesting to us, ranging from phenotypic plasticity to population divergence on islands, responses of range limits to climate change, and how behavior, physiology, and life history respond to the environment. So he did his undergraduate degree in biogeography at UCLA. He then did a PhD at the University of Montana, working on ecological and evolutionary determinants of incubation strategies in nuthatches. So he was working on birds at that time. He then went to do a postdoc on fish at University of California, Riverside, on guppies. And after that, he was for 17 years at Colorado State University, mainly working on aquatic insects. In 2020, he moved to the Norwegian University of Science and Technology in Trondheim. And now he's working on terrestrial insects as well. So he works on a lot of interesting stuff, and he's going to talk to us about some of those differences between organisms, between tropical and temperate environments today. So thank you very much, Cameron.
Okay, muchas gracias for the invitation to speak with you today. As Rob mentioned, I work on a lot of different kinds of systems and questions, but one of my longstanding interests has been this comparison between temperate and tropical environments, and why the tropics are more diverse and have more species than the temperate environment. And so I'd like to start with this painting of Alexander von Humboldt and his partner in crime, Ami Bonpland. And so in 1799, von Humboldt and Bonpland came to what is now Venezuela to do an inventory. And von Humboldt was really the first sort of scientist, we could say, to come to the New World and study things not as a colonizer, but as from a perspective of discovery. And if you read his journals, you see that when he arrives, he says, "Okay, we've arrived, and we're going to do an inventory of the species diversity here, and then we will move on." And 3 months later, you see he also writes in his journal, "We've been here 3 months, and we have only scratched the surface of all of the biodiversity here. There is more biodiversity in 1 hectare in this area than there is in all of Western Europe. And we could stay here our entire lives and maybe not completely quantify this diversity." And so in the process, von Humboldt was really the first person to describe this latitudinal pattern of increasing species diversity as we move towards the tropics. And this question of why, I should also mention, von Humboldt also noticed that there was interesting interactions between latitude and elevation. And so he had these beautiful illustrations of mountains in South America. This is Chimborazo in Ecuador in the Andes. This is the Matterhorn in the Alps, and this is a mountain in Lapland in northern Finland. And you can see that von Humboldt also noticed that there was an interesting pattern of zonation across these elevational gradients. And that the sort of pattern of species turnover also varied depending upon whether you were at a high latitude or a low latitude. And so there was much greater zonation in tropical mountains than there was in temperate mountains. And so this has led to this kind of long-standing and still somewhat unresolved question as to why is it that we see more species in the tropics? And so we can see over time there have been various publications putting forward various hypotheses, both for why we see greater latitudinal patterns of species diversity, but also across elevational gradients as well. And we know that at some level, these differences in diversity are related to climate. So if we look at species diversity in birds, you can see that in red here, in the sort of tropical regions of the Earth, is where most of the bird species diversity is. And if we look even more sort of focused on specific areas within the tropics, we see that mountain, especially wet mountains, like the wet areas in northwestern South America, are where the highest bird species diversity exists. So, so what is it about climate that might drive higher species diversity? Well, we could look a little bit more focused at some aspects of climate. For example, if we plot evapotranspiration versus number of species, we see that there's actually a very strong relationship. So evapotranspiration incorporates both temperature and rainfall. And again, you can see where these hot spots occur in the Andes, where you have very, very high species diversity, very warm temperatures, and very high rainfall. But we still have a problem in that these patterns are largely correlational, and it doesn't tell us why we see diversification. Why is it, for example, that there should be many species of birds in a tropical wet mountain area versus just one species that's very abundant? What is it about climate? What is the mechanism that might lead to reproductive isolation and the formation of new species? And so this remains kind of an open question, the mechanisms by which climate might result in higher species diversification. And so one explanation for how climate could promote speciation and diversification was put forth back in 1967 by Dan Jansen. So those of you who are familiar with tropical biology are familiar with Dan Jansen's name. He's one of the preeminent tropical biologists. And he provided one potential hypothesis for how climate variation across latitude and in combination with elevation could shape patterns of physiological diversity, patterns of dispersal, and associated reproductive isolation. And this paper, Why Mountain Passes Are Higher in the Tropics, inspired a lot of early work in comparative physiology in the 1970s, and it's often invoked as an explanation for tropical biodiversity, particularly along tropical mountain gradients. So, so before I explain what Jansen's hypothesis actually is, I kind of want to give a little bit of a side story here, which is this was a paper that I read in graduate school and I thought was fascinating and very inspirational. Borja had the same opinion, but it was also something that is not easy to kind of study, especially during a PhD degree. But later, when I started at Colorado State, I was at a conference with Ray Huey, and Ray was one of the few people who had actually published papers on Jansen's hypothesis and was interested in it. And I talked to him and I said, are you still interested in Jansen's hypothesis? Or, have you thought about it? He's like, I would love to get back into that. Let's start thinking about it more. And in combination with some good friends and colleagues, Paul Martin and Josh Tewksbury, we've wrote a few papers. These are two of my most highly cited papers that just started from a conversation. So I guess I would say to those of you who are early career graduate students or postdocs, you never know where a simple conversation might lead. This simple conversation I had with Ray at a conference kind of completely changed my research interests and trajectory. And so, don't underestimate the power of a simple conversation. Okay, so what was Jansen talking about? Why did he have this funny title of why mountain passes are higher in the tropics? And it's a little bit, can be a little bit confusing, but actually rather quite simple in its argument. And so basically what Jansen argued was that if you look at a given elevation in the tropics versus a given elevation in the temperate zone, at that elevation, because of the difference between summer and winter temperatures, you see a fairly large range of temperature variation at any given elevation. But if you go to the tropics, that seasonality is largely gone. So there isn't, when you're at the equator, there isn't a really big difference between summer and winter, at least with regard to temperature. And so the range of variation at any given elevation is much smaller in the tropics compared to the temperate zone. Now, if you were to move up, so here's this range of variation. If you were to move up then in elevation in the temperate zone or the tropics, what you will see is that in the temperate zone, it gets cold at all elevations in the winter, and it gets warm at all elevations in the summer. And so there's actually quite a bit of overlap in temperature between high and low elevation sites. So for example, you can imagine springtime temperatures at low elevation might be very similar to summertime temperatures at high elevation, say here in Spain. But in the tropics, this is not the case. It gets colder as you move up in elevation, and it stays cold. And you don't see this overlap in temperature. And so as a consequence, if you are an organism living at high elevation, you never see these warmer temperatures. And if you're in the lowlands experiencing these warm temperatures, you never see these colder temperatures the way that you would in the temperate zone. And Jansen said this has a profound effect on these organisms because if an organism were to disperse and try to move across an elevational gradient in the tropics, it would very quickly encounter temperatures that are outside the range it would normally see. Whereas in the temperate zone, that's not the case. And so he argued that dispersal should be much easier. So I like to visualize this as this kind of dessert that my mom used to make when I was a child. And I kind of see tropical mountains as these layers of, well, this is this kind of jello that we would eat, but you can think of these as bands of temperature. And so they're largely non-overlapping in the tropics. So what might the consequences of this be? So if we compare a high latitude site that goes from winter cold to summer warm temperatures, and there's this wide variation versus largely stable temperatures in the tropics, what Jansen predicted was that this should favor a very broad physiological tolerance, and this should reduce the cost of dispersal because moving across these gradients you stay within this broad physiological tolerance range. Increased dispersal should favor higher gene flow. Higher gene flow should reduce the potential for allopatric speciation because there's more genetic exchange between populations. In the tropics, we might predict the exact opposite. So at any given elevation, you see very little variation. This results in a very narrow physiological tolerance. This leads to increased costs of dispersal, physiological costs. This reduces gene flow between populations, and this could favor higher allopatric speciation. So the reason that tropical mountains are higher, according to Jansen, is not because they actually are necessarily higher from an elevation perspective, but they're physiologically higher, and they pose a greater physiological challenge to dispersal compared to temperate mountains. So the goal of today's talk is to basically ask, what is the evidence supporting or refuting Jansen's climate variability hypothesis? And to do this, we're going to look at temperate and tropical aquatic insects as sort of a model organism. So this is a big project that was an interdisciplinary group of people that worked on it. So stream ecologists, population geneticists, taxonomists, and this is sort of our EvoTrack team. And what I'm going to talk about today is some of the main assumptions and predictions that come from Jansen's hypothesis, working at a temperate site in Colorado and a tropical site in Ecuador. And so the first thing that I'll do is test this assumption that climate variability and temperature overlap is actually reduced in the tropics. So we can think of this as the climatic template that Jansen described. And then the first main prediction, which is that tropical insects should have narrower thermal tolerance, greater local adaptation, and reduced thermal acclimation, all predictions that Jansen made. And then, by extension, whether there's reduced dispersal in the tropics and whether this favors narrower elevational ranges. So is there reduced gene flow between populations? What is the pattern of species turnover and diversity across temperate and tropical elevations? And whether there's evidence for speciation rates being higher in the tropics. So let's jump into that. So first, we should ask, why streams? And one really important reason why stream insects are good models for this is because both temperate and tropical streams typically stay flowing all year round. So they may freeze on the surface, but they stay flowing underneath. And that means that the aquatic insects that live in these environments see the full range of temperature between summer and winter in both sites. So there's no diapause or dispersal or migration away from these winter temperatures. And also that stream invertebrates represent a major component of biodiversity. For me, this was a kind of a new area that I moved into, and I was fascinated by the communities that occurred in these environments. Whoops. So you have predators and herbivores and grazers and decomposers and just such a wide range of interactions that occur within these communities. And so despite this, it's also sort of an understudied component of biodiversity. And so it was another motivation for looking at these environments and these taxa. There's another component that I won't talk about today, which is that when we think about temperature and water, we also need to think about dissolved oxygen, because it also poses kind of a correlated aspect of the environment that might be very important. So we know that, for example, as temperature increases, the capacity to hold dissolved oxygen goes down. And so at the same time that dissolved oxygen is going down, warmer temperatures also favor higher metabolic rates in invertebrates. So the demand for oxygen is going up as the supply is going down. We've investigated some of this, but didn't find a lot. But if you're interested in this, please talk to me afterwards. Okay, so our study sites, as I mentioned, are in Colorado, 40 degrees north latitude, versus right at the equator in the Napo province in Ecuador. And we studied small tributary streams that were wadable, meaning that we could walk inside of them. So we didn't sample in very large rivers that were too deep for us to investigate. So the size and the flow and everything that we could control for between the temperate and tropical sites, we tried to maintain as the same. And so, so these were our sampling locations, and we did this across replicate watersheds. Okay, so first question. Do we see less overlap between temperature in tropical sites, like here in Ecuador, compared to Colorado? And the answer is yes, very obviously so. So here's our Colorado site. So if you put temperature loggers in streams, you can see that at lower elevation sites, it's warmer than at higher elevation sites in the summer. But as you approach winter, it gets cold everywhere. And temperatures approach freezing throughout the winter, with some spikes associated with warming events. But in general, it gets cold everywhere. But you can also see that there's quite a bit of overlap. Even at the higher elevation sites, you can see that they overlap with some of these low elevation sites. In contrast, if we look at the tropics, we see not much seasonal variation, and most of the variation that you see within a site is the difference between daytime and nighttime temperatures. And that variation tends to increase as you get to higher elevations. So especially when you get above tree line, the daytime temperatures can, even at high elevations, can be quite high, and the nighttime temperatures can be quite low. And so in some cases, the variation between daytime and nighttime temperatures at high elevations in the tropics approach some of the variation that can be seen even within summer and winter in the temperate zone. So if we look at the stream temperature range as a function of elevation, we see that range between tropical streams at different elevations tends to vary around 5 degrees, but in the temperate zone, most of the variation is occurring at low elevation. So at low elevation, it gets very hot in the summer and very cold in the winter. So there's a big range. But as you move up in elevation, that range of variation gets smaller. And so it's interesting because if we were to extend and go to very high elevation sites in the temperate zone, it starts to converge so that high elevation sites, both in North America and in South America, actually might experience the same range of temperature variation. So this climatic template is a little bit more nuanced than I think we originally appreciated. So in other words, when you think about a tropical site, you might think of a nice, warm, humid tropical forest. But it's also important to realize that right on the equator, we have very large mountains, and these sites basically are snow-covered and are essentially living in winter conditions all year round. So this is Mount Antisana in Ecuador. Okay, so we have this climate template. how does this shape the physiology of the aquatic insects? And so I'm going to highlight work by a former PhD student, Alicia Shaw, who's now an assistant professor at Michigan State University. So this is all her work that I'll be talking about today. So what Alicia did was she collected insects from the field. She acclimated them for 2 days in either their home temperature or a +5 degree acclimation treatment. And then she looked at the critical thermal maximum and minimum temperatures. So these are the maximum and minimum temperatures at which the insects are able to maintain function. So when they reach these critical temperatures, they sort of lose function. They lose the ability to right themselves, or they may start to exhibit muscle spasms. And so she did this across a wide range of the 3 main groups of aquatic insects: the Ephemeroptera, the mayflies, the Plecoptera, the stoneflies, and the Trichoptera, the caddisflies. And so the first thing she found was that in general, CT max is much higher for temperate species, which you might at first think, oh, that's interesting, because you would think the tropics are warmer and they should have higher heat tolerance, but in fact, this is not the case. But you can see that heat tolerance is going down in both temperate and tropical sites as you move up in elevation and the temperature gets colder. And you can also see that there's kind of, this slope is a bit steeper, and you start to see perhaps towards that convergence at high elevation that matches the climate variation. And if we look across the 3 main orders, consistently CT max is higher across all groups. And if you do a phylogenetic correction, we see the same pattern hold across all of our groups. If we then look at CT min, the cold tolerance, in Colorado, it's largely invariant. So at all elevations, because all these insects see very cold temperatures in the winter, they are all equally cold tolerant. But then look at the tropics, really, really big difference. So these low elevation tropical species that really rarely see cold temperatures have very high CT mins compared to their high elevation counterparts. So again, you can see here, especially for cold tolerance, high elevation temperate and tropical species are actually quite similar to one another. And the big difference occurs at lower elevations, not at high elevations. If we put these together, what you can see in blue are the temperate species, so they have lower cold tolerance, higher heat tolerance, and so they encompass a wider range of temperatures. So we would say they have a broader thermal breadth compared to their tropical species, which are inside the full range that we see in the temperate species. So what's interesting is if we plot this thermal breadth as a function of elevation, again we see that the really big difference occurs at high elevation, but at low elevation, but at high elevation we start to again see this convergence. So the thermal breadth, the range at which the full range of temperatures that these insects can tolerate is actually not that different for high elevation temperate and tropical species. And if we then plot the variation in stream temperature range versus this thermal breadth, what you can see is that it's not so much whether you're in the temperate or the tropics. What really matters is how much variation do you experience. That seems to be the main predictor of the thermal breadth. So this was really, really interesting for us. And we also wanted to know the capacity of these insects to exhibit acclimation, so a kind of a thermal plasticity. And so Alicia compared the home stream acclimation to the +5 degree thermal acclimation treatment. And what she found was that for mayflies, the acclimation effect resulted in a higher CT max. So they were the temperate species were able to have an increase in their CT max. This is for low and mid elevation, but the tropical mayflies did not show an acclimation response. And that's kind of what we predicted, because if you don't see much variation in temperature, like seasonally, then there's not very strong selection to maintain an acclimation response. Interestingly, in stoneflies, shown here on the right, there was not much of an acclimation response for either temperate or tropical species. And this is interesting because stoneflies are a temperate group. The Plecoptera have a temperate origin, and in geologic time have only recently invaded and colonized the tropics. And they tend to occur in these high elevation, cooler environments. So kind of matching their temperate environments. So that might be one reason why we don't see this response in the stoneflies. But these results are kind of consistent with other studies of ectotherms that show tropical species tend to have a weaker acclimation response and less plasticity compared to their temperate counterparts. Okay, so Alicia also wanted to know what's happening in between CT min and CT max. So far I've been telling you about the difference between these two in the thermal breadth, but these are sort of the extremes. And so what happens in between? And so she was interested in looking at how the metabolic rate changes and how sensitive is the metabolic rate to temperature in temperate and tropical species. And so there hasn't been a lot of comparative work, but we predicted that the narrow range of variation in the tropics should favor a steeper and a higher peak in the tropical species metabolic rate profile. So a rapid increase and a rapid decrease. And then in the temperate zone, we should see a broader sort of difference between the CT min and max and a more shallow kind of rise and fall with respect to the metabolic rate. And so to do this, she did closed respirometry using this microrespirometer from a company in Denmark called Unisense. So you can see here the mayflies in a little 4 milliliter chamber with a little stir bar underneath. And she looked across a wide range of temperature from 5 to 25 degrees, total of 370 total insects from 3 elevation and from the 2 latitude sites. So this is a lot of information, so I'm going to have to walk you through this. But what I'm showing you here, let's start on this side. So these are the metabolic rate profiles as a function of temperature in the temperate zone here on the left and on the tropics on the right. And so if we start with to orient you, here is the oxygen consumption, so a proxy for metabolic rate. You see that it rises. The arrow represents the peak, and then usually followed by a decrease. So we see this peak. So these aren't like these beautiful, nice curves that you see in the textbook. They're a little bit more messy. But in general, you can see this rise. And then in gray, we have the thermal variation that the insects actually experience in nature. And so what you can see is it's not perfect, but typically the peak occurs just outside the range of variation. Not so much here, but you can see it at the higher elevation sites. And that's also the case here in the tropics. So once you move outside the typical range is when you start to get this peak. The other thing to point out is the red bars. So in red are cases where the insects showed visible signs of stress. And either in terms of like muscle spasms or other kinds of like proxies for stress, the inability to like hold on to the mesh that we put in there. And you can see that there's no red in the temperate zone, but in the tropics, for example, at low elevation, 5 degrees is stressful, and 25 degrees is also stressful. And then if you move up in elevation, higher temperatures are stressful, and even more so at our highest elevation sites. And so as a result, what we see is that if we put all these together, tropical species have a steeper increase in metabolic rate and often a steeper decline in metabolic rate as a function of temperature. So in particular, when you move outside of the range, these narrow ranges that the tropical species encounter, you start to see a greater sensitivity, the metabolic rate increases more rapidly, and you see behaviorally, you see more stress. And so if we plot these as just the peak values for the different elevations, we see also the tropical species have higher baseline standard metabolic rates. And so unlike other kinds of performance measurements, a really high metabolic rate just in response to temperature is not necessarily a good thing. It means that you're expending a lot of energy just because of temperature. And that means that there's less energy available for other activities and other processes. So we could say in physiological speak that we observe higher Q10 values in the tropics. And so at any given temperature, this means that tropical species are expending a lot more energy. This could trade off with growth and reproduction. We have a manuscript on this where we show that tropical species seem to experience a stronger trade-off between metabolism and growth. And this has implications for what's happening to their food availability. So if you have higher metabolic rates, especially as we move towards a warmer future, is food availability going to keep up with these higher metabolic demands? So at least from the physiological perspective, we see some general support for Jansen's hypothesis. We see greater thermal breadth in the temperate zone. We see greater sensitivity to metabolic rate. We see these higher peak metabolic rates. So altogether, it suggests that tropical species are adapted to a narrower range of temperatures as the climate variability hypothesis put forward. What about then the remaining predictions? How might this affect dispersal? How might this affect patterns of diversity across elevation gradients? And how might this approach speciation? So let's look at those now. So now I'm going to talk about a former postdoc, Nick Pilato, who worked in Kelly Zamudio's lab, who did a RAD-seq study looking across elevation gradients in both the temperate and tropical areas to get a proxy for how much gene flow and how much genetic exchange there is among different populations in the temperate and tropical environments. And if you look at, for example, one measure of genetic differentiation, so this is using pairwise FSTs, we see that there's higher FST values, so higher differentiation between tropical populations compared to Colorado populations, suggesting more gene flow in the temperate zone. If we look at the number of migrants per generation, another estimate of gene flow, again, we see higher migrants in Colorado in the temperate site. So collectively, more, and there are many other studies that have also found reduced gene flow between populations comparing temperate and tropical environments. So these results are consistent with what others have also noticed. Another graduate student, Brian Gill, then looked at patterns of diversity and distribution across elevational gradients. And so the sort of workflow that Brian did was tremendous amount of work. So he collected larvae and adults using standard sampling and collection at different sites. He then cataloged and recognized taxa based on their morphology initially. So kind of the best morphological taxonomic unit that he could. In Colorado, where the species are better known and better described, he could usually get to the species level, but in Ecuador, he could usually only get to the genus level. All of those, at least a subsample of these specimens were then sequenced for CO1 to get a DNA barcode. And using the distance, we delimited them by species. And then he did phylogenetic analyses to make phylogenetic trees based on this one genetic marker. So the big question was, is there greater species diversity in tropical mountain streams? And this was actually an open question, because if you go to a stream in the tropics and you look at the aquatic insect diversity, it's not like looking at birds, where you see much greater diversity around you. It looks more or less the same in both environments. So the question was really an open one. And so what Brian found was actually very interesting. So what I'm showing you here are the three main taxonomic groups: mayflies, stoneflies, and caddisflies. And then in orange, these are the descriptions based on morphology, and then in red, the description based on barcoding. And what you can see, for example, in the mayflies is that our morphological descriptions between the two sites is much less than what we pick up when we barcode our samples. So there's a lot more cryptic diversity, but this cryptic diversity is much greater in the tropics than it is in the temperate zone. For the plecopter, which I said earlier is a temperate species, there is actually more plecopter species in the temperate zone than there are in the tropics. But then look at the tricops. There's a lot of cryptic diversity here. And what was really interesting for me was one of the mayflies that we commonly studied, we found at almost all of our sites across the Andes. And morphologically, it looked identical. We couldn't see any difference across all of our sites. But when we barcoded, every population was highly genetically divergent from all the other populations. So the diversification isn't happening morphologically. It seems like it's happening physiologically across these different sites, where they are becoming more locally adapted to narrow sort of temperature environments. As a consequence, when we look at the morphology versus the DNA barcode species, we see that the range occupied by the species in Ecuador compared to Colorado is much smaller. So there is more turnover of species across the elevation gradient, which is exactly what Jansen had also predicted. And this pattern, again, is robust to any phylogenetic signal in the data set. So tropical species have narrower elevational ranges that they occupy. So what about speciation? So what Nick did was he used this binary state speciation extinction model, this busy model that Wayne Madison and others have developed. And I have to confess, this is a bit of a black box to me. What this method does is use maximum likelihood to calculate the probability of basically seeing the pattern on the phylogeny given some assumptions about speciation and extinction rates. And you will have to, I will defer to Nick about the details of the method. But using this method, which others have used as well, what you see is that the speciation rate in Colorado relative to Ecuador seems to be much slower, and that you see more branches forming in the tropics, consistent again with this idea that reduced gene flow and reduced genetic exchange is happening. I have to say that this pattern of diversification across elevation gradients in aquatic insects is not necessarily universal. We've also published on birds, and the evidence for birds suggests this is not the case, that most speciation in birds is not happening in situ across elevation gradients. But that's a separate topic that we can talk about later. So for this first part, generally, a lot of evidence in support of Jansen's hypothesis in terms of the climatic template, in terms of the physiology, the dispersal, the turnover of species across elevational gradients. And also that these patterns are sensitive to looking at cryptic species diversity, not just the morphological diversity. But there's a big caveat here that we recognized early on in the study that was something that bothered us, which was that when we look at the distributions across the elevational gradient, the thermal breadth actually doesn't do that good a job of explaining the patterns of distribution that we see. And for example, if we look at a high elevation mayfly, the CT max, the environmental temperature is 6 degrees C, but it can tolerate quite warm temperatures, up to 26 degrees before it reaches its critical thermal maximum. So why doesn't the species move down in its distribution? And similarly, low elevation species, especially like in Colorado, they can tolerate quite warm, quite cold temperatures. Why don't they move up? Perhaps species interactions might play a role in this. And so we became interested in particular the predator-prey relationship between stoneflies and mayflies, and whether that might overlay another factor that might influence their range limits. And so we've been thinking about this a lot in my group. So Alex Marrow, who's here, had a nice paper a few years ago where he argues that we often in comparative physiology make these performance curves for single species to understand their responses to temperature, but we rarely do this for interacting species to understand how interacting species performance changes across different environmental gradients, for example, temperature or salinity or other things, and whether at different parts of the gradient, one species might have an advantage over another. And so, for example, in our mayflies, if there's some pattern of performance, we might predict that as performance starts to decline, they might become very vulnerable to, say, predators or competitors or other interacting species. And so what Alicia did was look at swimming performance of mayflies and stoneflies from different elevations in both the temperate zone and in the tropics. So this is a low elevation mayfly, low elevation stoneflies. The gray lines are the tropics. And you can see that there's quite a bit of variation. In general, we see that the tropical species tend to have more kind of a peak in the mayflies, but that the stoneflies seem to kind of maintain variation across a range of temperatures. So it was a little bit unclear who has the real advantage here. And And so to get at this more experimentally, what Alicia did was simulate what would happen if a low elevation species moved up or a high elevation species moved down. So to do this, she simulated a high elevation environment, 6 degrees, with a resident stonefly predator and resident mayflies, and then in black, low elevation mayflies that were brought up. And we could tell them apart because of the number of little extensions off the back of the body. And then same thing for low elevation, residents within simulating high elevation species moving down and to see what would happen. So the predators were starved for 24 hours. The mayflies were acclimated for 24 hours, or sorry, 48 hours before the interactions were allowed to take place. Interestingly, and consistent with what we saw before, acclimating for 48 hours in these temperatures was much more stressful for the Ecuadorian species. So we saw a lot more mortality compared to the Colorado species. So this is just not independent of the predation, just living in these kind of warmer or colder conditions, regardless of whether you were simulating moving up or simulating moving down. And interestingly, regardless of whether you were high or low elevation, the residents tended to have lower mortality compared to the transplanted individuals across both sites in temperate and tropical environments. So if you put these together, what you see is that total mortality in Ecuador is higher, and it's even higher for the transplanted individuals compared to the resident individuals. So being a resident, you have some advantage, and this difference between Colorado and Ecuador is not so much because the predation rate is necessarily higher, it's because the combination of the physiological stress and the predation results in this big difference. And this is important, I think, because there have been several hypotheses and arguments going all the way back to Darwin that biotic interactions are stronger in the tropics or at low elevation compared to high elevation or temperate environments. And I think those conclusions often confound the biotic effect of the predation and the physiological response. And that it may not be one or the other, but the combination of the two that often is what people are actually measuring. So, you know, how much does this pattern reinforce the patterns of elevational distribution that we see? You know, we can discuss that. That's an important question, I think, to ask. So overall, many of the assumption and predictions from Jansen's climate variability hypothesis are supported, but a lot of these still remain correlational. We need more experimental work to kind of get at this, especially the generality of these predictions in combination with biotic interactions, which Jansen did not talk about. And we have to also put these in a kind of a conservation, these results in a conservation context, because climate warming and extreme events are happening globally. And it seems that in the tropics, where the species diversity is the greatest, where the thermal sensitivity is the greatest, is also potentially where there may be the greatest risk. And this variation also is occurring across elevational gradients, so the responses may not be the same when we talk about temperate versus tropical. We have to be careful about also what elevational distributions we're talking about. So I'm not talking about this today, but as Rob mentioned, we're now working locally here, just outside of Madrid, on a kind of a spin-off project now looking at terrestrial insect species across a latitudinal gradient in Western Europe. And so we have the team here that's kind of touching on a lot of the same sorts of questions that we did with aquatic insects that are now being done with terrestrial species. And so hopefully we can come back here in a few years and share the results from these results this study with you. And so this is a team effort from a lot of people, a lot of funding sources. And I also want to just make a plug. I also am a co-host for a biology podcast called Big Biology, where we talk to other scientists about their research. And if you're interested in talking with us about your research, please talk to me and I'd be happy to see you on Big Biology. So thank you for the time, and I'm happy to answer any questions.
Great, thank you very much, Cameron. Okay, questions? Okay, I'll start it off quickly.
Okay.
So why is CT max higher for temperate species than tropical species?
Well, we're not 100% sure, but we think it's largely because it actually gets warmer and in Colorado. And so many of the sites are open canopy sites, so it can get quite hot in the summer in Colorado. So easily, temperatures can get as high as like 40 degrees. But certainly in the mid 30s is not uncommon. In the tropics, a lot of the streams, although they can be open canopy, the canopy has a moderating effect. And so it's interesting, like if you go, again, we think of the tropics as being hotter all the time, but if you're under the canopy in the forest, in the shade, it's actually not as bad. And there's been some work showing, like with lizards, that species that live under canopies tend to be more sensitive to temperature than species that live out in the open. And it's the species that live under canopies that tend to be showing greater declines. So I think it's not universal across all elevations, but that's our best guess for why the CT max is greater.
I've got one from Diego here.
Yeah, thank you. Super nice talk. Thank you very much. I was wondering to what extent other key physiological functions like reproduction or immune function could explain best this difference rather than just...
Yeah, that's a great question. Aquatic insects are really good for certain things. They're also very bad for other kinds of questions like this. So, I would love to be able to bring these into the lab and look at more correlates of fitness, like growth and reproduction. And I think those are really, really important questions, because when we look at thermal sensitivity, it's not the same for different traits related to fitness. I saw a talk by Dave Buckwalter a few years ago that was fascinating and also very disturbing, where he showed performance curves for growth, reproduction, survival, metabolic rate, and the peaks were all at different temperatures. And so depending on which trait you were looking at, you would get a very different conclusion as to what is the optimal temperature. So that's I think very important, and it remains a big gap.
So there is not a single proxy for...
Yeah, we have to be very careful about how we interpret them, but yeah.
Hi, very interesting and nice talk. I'm interested in what are you doing, and I have trouble with the acclimatization in your experiments, right? Because what about you're taking the adults or the larvae, right? Or the nymphia?
The larvae, yeah.
Yes, the larvae. And you're taking out and then putting them in some hours, like 2 days, something like that, to acclimate, right?
Yeah.
So in the idea is that they will acclimate to some new environment, but in fact you are just stressing up they from their home, right? So what if you put them to develop the entire development process into one environment? And the other thing is, what about the variation in temperature? I know that into water is less than in the land, but water also have variations, right? I think so. So what about those variations and not just being 6 degrees there all the time, stressing up, and stressing up, right? So what about that?
Yeah, so, the question of sort of keeping any organism at a sort of a static, constant temperature versus a more natural, dynamic temperature is something that has been debated a lot. And in fact, if you mimic the variations, so work by Katie Marshall and Caroline Williams have shown that that can matter, especially for CT-min. That does have some influence. So we recognize that as a problem. The 48-hour acclimation is partly just from a logistical purpose. It's not so stressful for the temperate species. It is more stressful for the tropical species. So we definitely see that difference. So it's consistent with what we see, at least in our other measurements. We also know that the longer they have to acclimate, typically the better they acclimate at. But it is this fine line between, as you mentioned, sort of adaptive acclimation versus stress. Because when we keep them in these acclimation trials, we also don't feed them because we don't want to confound the cost of digestion with the effects of temperature. But there's only some amount of time that you can go without eating before it also becomes stressful for you. And so, these are just logistical challenges, that are common with these types of experiments. And so, we do the best we can, given these constraints. But I guess I would say we have to start somewhere and first kind of get these patterns, and then we could follow up and see, how will the results change with longer acclimation times? How will it change if we fluctuate the temperatures instead of keeping them static? So, yeah.
Well, first, I want to thank you very much for putting all those beautiful pictures of my country. My heart really warmed up when I saw Mount Cotopaxi in all its glory there. It's very close to my house. Well, first that. Second, it's kind of like a comment. I don't know if you have any comments to that. Is that yeah, all these studies with these aquatic insects, especially related to the last part when you were talking about biotic interactions and more the nitty-gritty of the ecology of the species, is that when you study the community in the water, it's just half of their story, because the adults are doing all sorts of stuff outside the water, right?
Yeah.
So who knows what is going on with the adults outside?
Yeah.
And that really struck me, because you were telling us that at the end, yeah, this is all very interesting, but we still cannot plot the distributions of the species given their physiological thermal limits. Mm-hmm. So this just kind of like a comment. The other one is, to what degree have you thought of all these patterns, but not from the perspective of elevation, but from the perspective of latitude. Because I often think, at least in the tropics, the elevational gradients are kind of like a latitudinal gradient, like vertical, right? You can see the whole gradient from the from lowland rainforest to permanent glaciers, right, at the top. So do you know if kind of Jansen's ideas also occur across the latitudinal gradient? I guess the prediction would be that the latitudinal ranges of a species should become narrower, closer to the equator, and become broader, closer to the poles.
Yeah, and I think that's more or less what we see. And as you point out, at where it kind of starts to break down is at the very high elevation sites, because then the top of Antisana is not that different from the top of the Rocky Mountains, with permanent glaciers and, very cold temperatures all year round. One thing back to your comment about the life cycle of the aquatic insects, for those of you who aren't familiar, most of these aquatic insects, 99% of their life cycle is spent as larvae in the water. And in some cases, like for some mayflies, the adults emerge for just a very, very short amount of time where they reproduce. And that is actually potentially when some of this dispersal is happening. So if you think about the streams are moving water down, and so over time, maybe everything should be at low elevation. So it's thought that some of the adult dispersal must be moving upstream to counteract this. But working with adults that are very short-lived and doing these types of experiments is another gap in our knowledge. So, people who work on flying mayflies and stoneflies and caddisflies, like this is one of the most important questions I think that you could ask is how does temperature affect their flying performance and their patterns of dispersal? Because really we know nothing about it, and it's a very important gap in understanding these distributions. So I would say that most of the interactions, though, still probably occur underwater, and in the stream ecology literature, the prevailing idea among community ecologists is that biotic interactions are not that important because streams are very disturbed sites, so they're always under kind of non-equilibrium conditions, and they're subject to flooding, and so you don't have a lot of stability, and so competition and predation are not as important as they would be in terrestrial environments. But I'm not sure that that's actually true. I think you do see a lot of, these stoneflies, when you watch them, they're hunting all the time. They're like lions and they attack, and the mayflies, to escape, will sometimes like release from a rock and then just drift to escape the predators. So whether they then slowly make their way back upstream, we don't know. But certainly, I think, to ignore biotic interactions as playing some role in the distribution of species would be wrong. And I think it's often presented as like it's only abiotic effects or only biotic effects, but it has to be the combination. And so, in the bird literature, people like Ben Freeman have looked at a lot of bird distributions and argued strongly for biotic interactions and competition. And I think that there's still some evidence that it's not just biotic interactions. There's also some competition going on as well.
Just right about that, I was thinking, what about the top predators like Odonata and Dobson flies, like Megaloptera? Have you made some work on those orders?
We haven't, but there's obviously many other in addition to Dobson flies, you also have dragonflies and what's the other relation?
Are there flies? No.
No, what's not damselflies.
Damselfly.
Yeah, and those are like the scary predators. Really, like when you see them, they're eating like everything. So so their numbers seem to be like some places they can be very locally abundant. I think where the stream flow tends to be more slow, and certainly like in ponds and lakes, they can be very abundant.
Sorry, but for example, Megaloptera larvae are really top predators that
yeah.
eat all those Plecoptera and mayflies and everything. So is the same happening for these top predators, like city max and everything?
I'm sure it is. We haven't looked at it yet. I mean, we focus primarily on the three main taxonomic orders of aquatic insects, but yeah, there's many other invertebrates out there, and in general, aquatic insects are understudied compared to terrestrial invertebrates as well.
Any more questions? Okay.
What's that? Okay. Okay.
Okay. Yeah.
right.
Well, thanks for the rest of the questions are going to be down at the kiosco where we're going to take Cameron and his group, or come and speak to him now. So thanks everyone for coming and watching online, and thank you, Cameron.
Thank you.